are the two sensors. I assembled these here in the lab. We put the electronics together and soldered it up. This right here is the head. You can see it's got four. Maybe if I back up a little bit, you can see it's got four diffusers. It's got four diffusers pointed up, and it's measuring in four bandwidths: 800, 676, 568, and 532 nanometers. And then the other one, the other end of it, is doing the same thing, only it's pointed down at this vegetation, which is a sedge, Carex bigeloi mostly, but there are some other sedges and some grass in there as well. So we have these two sensors, and they're logging. The photodiodes, of course, create a current. The current comes down here. There's a op-amp in here that amplifies the output. This is just a junction box for the hobos. The hobos are data loggers. That each one has four channels, one for each one of the bandwidths. And so we log this, and by comparing the upward and the downward irradiance, the irradiance coming down, the reflectance of the vegetation coming back up, by comparing those two things, we can determine how much of the light is absorbed by the vegetation. This is, of course, a solar panel which is driving this sensor array, and this is a spectrometer here, a spectrophotometer. And early in the year, of course, the leaves are not open at all. So you have a very low, what's called a vegetation index, which is a ratio of the reflectances. And you have a very low vegetation index. And then suddenly, it begins to climb rapidly as these leaves open. The same thing occurs for evergreens as well, because they green up, but those leaves are already there. So you have a more linear response from the evergreens. And then this pipe is a mini rhizotron. The pipe goes in about 1 meter into the ground. You only see about 1/3 of that sticking up. And that pipe is clear. And a tiny video camera slid down the pipe. It's carefully calibrated as to the position of the camera so that you can actually look at particular areas of the sidewall of the clear pipe. And you can see the root growth, so you can track root growth with a video camera. So that's called a mini rhizotron. That's a control plot there. Over here, these are micro lysimeters. They look like little syringes, which is what they are, and they're sucking up soil moisture, soil water, out of that tussock. Okay, so you see that they've got the same setup times 10. It's just the number of replicates for this experiment. And somebody's collected some caribou vertebrae here. I've seen an Arctic fox up at this plot right here. There's quite a few birds. A lot of the birds have left because they're migrants and they've headed south. There are ground squirrels in this area, Arctic ground squirrels. Pretty good sized squirrels, and they hibernate, and the females are underground now. The males are still up and about. And then there are voles, which are smaller, also rodents. The voles actually don't hibernate. They are subnivean. That means that they live under the snow. This is a nice new footbridge that was put in. It's about 5 feet wide. It was put in over Inlet Creek. I usually stop and see what's in the water here. The grayling here. They're abundant in this area of Inlet Creek. These fences are about a little over 8 feet tall, They were built in about 1995, and they were part of the International Tundra Experiment, ITEX. So there's two of them here, this one, and look at all of this Arctostaphylos. This is bearberry, alpine bearberry, in the snowdrifts. The snowdrifts, of course, get to be about 8 feet deep here near the fence. Now, right next to the fence collects heat and acts like a radiator, and the snow melts and doesn't collect at the fence itself. These are the open top chambers that are put out. There's 40 or 50 of these chambers that get set in different locations. This lichen is called, I'm pretty sure this is Porpidia. I'm not sure the species name. These lichens grow very slowly on rocks. It can take centuries to cover a rock like this. So this is a crustose lichen that adheres closely to the rock, and the black spots on it are the fruiting bodies, the spore-producing bodies. The lichen is a fungus, and these spore-producing bodies are called apothecia. If we can look at them in a magnified view, they're somewhat cup-shaped, or they split open and release spores. It's one of the ways that lichens reproduce, being a composite organism composed of fungus and algae. They have several means of reproduction, not only by spores but also by vegetative propagules. Well, we've been walking for a few minutes up the hillside here, and I'm looking back at Toolik. This is Toolik Field Station, as seen from the hillside up above Toolik, and it's right on the end of Toolik Lake. Now, these large chambers here are not actually open top chambers. These are being used by some people from Duke, and they're doing a pretty interesting experiment here. It's they're studying wolf spiders and their position in the trophic web of the tundra, and how they're affected by advanced snow melt or climate change. So these large chambers that are about 6 or 7 feet across, some of them are enclosures to keep the spiders in, and others are exclosures to keep the spiders out. Well, we're up above the ITEX, the moist ITEX site. That's that moist fence in the background with the wind turbine beside it on the right hand end. And this area was used about 10 years ago roughly to do fertilization experiments in the tundra. This is the same dominant tundra here, the moist acidic tundra. The moist acidic tundra is actually the main vegetation type in the Arctic. It constitutes 26-27% of the Arctic is this tundra type that we're in right now. And there's quite a few mushrooms around here. This is a rusula. They're bright red. This one's sort of bleached out a little bit, but there are also boletes up here. This is one of the boletes that I was telling you about. This might be a king bolete. These are probably edible. Let me see here. I'm gonna turn this one over. They're pretty much spent, and see, you can see it doesn't have gills, so this is not a gilled mushroom. This one has pores that release the spores through the pores, and this one is a member of the Boletaceae, which makes it a bolete. As I said, it's probably a king bolete. Well, we're at the moist acidic tundra fence. This snow fence with its OTCs, they're stocked for the winter. We take them out at the end of the season, and we stack them in a few locations here. The wind turbine that powers the site is over here. Look how quiet it is today. No breeze. It's rare to see that thing without it spinning. And anyway, I spent most of last summer working on this site, correlating snow depth, which runs from about 8 feet deep near the fence. It stays that height out to about 20 meters, and then it begins to taper down, and the fence extends out to about 70 meters. So it's divided into different zones based on the snow depth, whether it's low, intermediate, or high. And then to the right of the snow fence, outside the drift zone, up here where there's a smaller met station, this is the control zone. So I did the same thing last year that I did this year in these areas, measuring the reflectance of the vegetation, the CO2 exchange, and species distribution, mainly those three things. This is the site I showed you from further up on the hill. This is an automated spectral reflectance site. These are solar panels here. The large boxes are for the storage of the batteries. The solar panels are trickle chargers for those batteries. The batteries run all of the sensors for this unit over here, the one on the tower itself, and the instrument under the blue tarp. This instrument under the blue tarp is a spectral analyzer that's pointed down as well as up. It's got measuring incoming radiation, solar radiation, and reflectance off of the vegetation. It's set up in this site because it transitions from the dry heat, the very low stature vegetation here. The cable system is strung across the transition zone between the dry heath, and you can see the transition zone right there, sort of marked by that line of shrubs, into the moist acidic tundra. That's the other tower over there. This is sort of like well, you might call this the new way of doing things. It's looking for automated systems. It wasn't actually possible to do these a couple of decades ago. So these automated systems are a preferred way of doing it now because the data can be logged. This data can be automatically put online. There's a wireless connection here that takes this data from the site to the internet without human intervention. This is part of the Arctic Observatory Network. Toolik is part of a long-term ecological research station, and the research network, it's all over North America. I don't know exactly how many sites there are, 20 or 30. And so Toolik is part of that network as well. That's the LTER sites. Toolik's a long-term ecological research station.